Hi, everyone. Welcome to our March meeting for SNHS. Remember that any important updates that we mention in this meeting can be found on our website at www.frhssnhs.org.
Oh, that's me.
All right, so here's what we're going to cover in today's meeting. We're going to start with some announcements. We have an update for fees for SNHS. Also, our t-shirts are here. So please, if you ordered one, pick those up and pay for them. We have an announcement on honor cords for our seniors in SNHS, and then we also are giving an update for those of you who applied for the board. We have more volunteering opportunities for you as well, and those include trivia night, applying to internships, summer programs, and scholarships, this week in science submissions, science memes, and remember to always check the website again for important updates. Anything that we mention here, you'll be able to find on the website.
Okay, so for the people who applied during the second wave of applications, so those are for the people who applied after, I believe it was September, the fees will be posted this week for you guys to pay, and please pay them as soon as possible.
All right, honor cords. So this is for all you seniors. To be eligible to receive your honor cord at this graduation, you must have attended at least 2 of the 4 meetings. We're listing the requirements a little bit. And if you'd like an honor form, please fill out that form that is hyperlinked on the PowerPoint by April 19th, or you will not be receiving a cord in time. Hope to see you with one at graduation.
Okay, so super exciting t-shirt update. Our t-shirts just got here. So you need to bring a proof of payment. So just like print your screen when you pay on school pay, and you can also get a receipt from the fees window. And just bring that to N112 during advisory or N102 during any other time to pick them up. And the fee is on school pay, or you can pay it at the fee window.
So for our 2021 to 22 SNH board applicants, thank you for everyone who applied for your board. Your time was really appreciated. For all those who did apply, make sure you check your emails throughout the week for your decision. And if you have any questions, feel free to DM me or email me or anyone else on the board, and we'd be happy to help you out.
All right, we are going to go through some volunteering opportunities now. They're mostly the same. If you haven't caught up on volunteering yet, please know that there are plenty of things for you to do.
So the first remote volunteering opportunity for you guys is actually the Falls Ridge Remote Trivia Night, which is happening April 3rd from 6:30 to 9:30 p.m. And just participating and signing up for this event will give you 3 hours that you can contribute to your just total hours. And to sign up, just visit this link at the bottom of the page at trivia.itsryan.org, and it's $9 per person, and you can sign up with teams of 3 to 5. So it's a great way to win prizes and have fun, and all the proceeds from this event are going to go towards the Make-A-Wish Foundation.
Okay, so again, if you guys are interested, you should definitely apply to some summer programs. I myself did one and I loved it. It was one of the best experiences of my life. Even with COVID happening, I did mine at NYU and it was just amazing. So if you have the time, make sure you apply, and you'll get 3 hours just for applying and another 2 if you get accepted to the program. And make sure to follow that link at the bottom if you want more information on some really excellent ones. Once again, if you are interested in signing up to do your own TWIS, just send me a 2 to 3 minute video of yourself presenting your topic before our next meeting, and we will enter it into our video. And finally, if you really can't find anything that interests you in the volunteering opportunities, you could also just send us 4 memes. So 4 memes for an hour, and the more interesting, the better. And if you make them yourself, bonus points. And again, if you're still lacking in hours, we have over 20 opportunities for you guys. Or you could just submit like 20 memes if that's really what you wanted to do. We've tried to make it really easy, and if you guys don't have the hours by the end of the year, we can't invite you back next year. And now for This Week in Science. We're going to start off with supercomputers and quarks. So this has been kind of a recent development that's built on the standard model for particle physics, which deals with how the elementary particles that make up subatomic particles interact with each other in terms of electromagnetic, you know the thing, and a couple of other things, but not gravity, which is the big problem with the system. The model at the moment is that it doesn't include gravity. So the standard model is really highly tested and it's very successful, except for the fact that it doesn't include gravity, which is a huge hole in the theory at the moment. So the other big thing that goes along with not understanding how it connects to gravity is that we don't know how these particles interact with each other. And so actually observing these particles requires massive amounts of energy. Think like the electron-ion colliders that you know of. Those massive buildings that require so much energy, and even those are not cutting it at the moment. So the solution for now is supercomputers. And so what they're doing is they're creating these simulations of these elementary particles that are almost physical, they say, which means that they have mass that's so tiny in these simulations that they're almost not actually mass, which is an interesting thing to think about. And so basically they construct a space-time grid on which the quarks can interact with each other using the properties that we've already observed from them in experimental observations. And as these simulations and supercomputers improve, we'll get a better snapshot of how these elementary particles interact with one another and hopefully fill in that gap with gravity. And so that should help connect and kind of advance and finalize what we know about physics, which is really cool.
Okay, so my This Week in Science presentation is on a new potential cure for neurodegenerative disease as well as other degenerative diseases. So basically the idea behind this new cure is, what if there was a way that doctors could transplant healthy neurons into patients that are living with neurodegenerative diseases such as Alzheimer's or Parkinson's disease, for example. So researchers at Northwestern University have discovered a 3D printable biomaterial that can actually mimic brain tissue that has degenerated due to neurodegenerative diseases. So the material that is being created is made up of molecules that react in the same way that proteins in your body would react, and they combine into superstructures that are, it's basically like a porous material that mimics tissue. And again, so these superstructures have bioactive properties that allow for tissue regeneration. So the material really mimics what you would find in human tissue. So once this superstructure material has been translated into the brain, in this case, it uses existing factors. In this case, it's called brain-derived neurotrophic factor to stimulate neuronal growth and essentially create new healthy neurons that have degenerated due to Alzheimer's or Parkinson's or other diseases. So this has a lot of implications, not only in the brain and for degenerative diseases, but also in replacing heart tissue or lung tissue or a lot of other organs in your body that have degenerated due to some disease or disorder. So these diseases are traditionally really hard to cure. Obviously, there's no good cure that we have yet for Alzheimer's and especially other neurodegenerative diseases. So this is really potent in potentially finding a new treatment for those disorders.
Cool. So I'm sure you guys have seen the new picture that's been floating around of the new black hole that displays those little lines are all the magnetic fields. And so it's kind of so it's obviously the first time that astronomers have seen these, and it kind of demonstrates the glowing hot gas that's around the supermassive black hole that is, of course, the center of the M87 galaxy, which is the same one that the famous picture that's taken in 2019. And they basically affect the black hole consuming matter, and it launches plasma jets thousands of light years out, which is like pretty much a really hard to imagine distance. And the jets have been measured before, but it hasn't really been known until this point how it's really been happening. But the same observation as what was used taking the 2019 picture was used, but polarization, which basically measures the orientation of the light waves, let the structure of the magnetic fields be traced. So it's kind of using the polarization allowed the picture to be developed with the lines, and the magnetic fields also affect the black hole by pushing against the material that's kind of spiraling in, doing what a black hole does, and it resists the pull of gravity, and which also leads to the conclusion that these are pretty incredibly strong fields, maybe the strongest out of anywhere in space that help push against the falling gases.
Okay, so my twist is about bunny handstands. There were researchers looking at this in multiple universities in a specific breed of rabbit called Sauter d'Arfort. A genetic mutation has caused them to do handstands instead of hopping. The gene responsible for this is called RORB, and it's similar to a Hox gene in humans in that it helps a fetus develop. And so in rabbits, this gene helps develop nerve cells in their spines to coordinate movement. And normally the unaffected gene would help rabbits coordinate their hopping movement. And as you can see, the effects are that changes into a handstand. And the specific effects of this gene are not really known to be only nervous or have more of a widespread throughout the body sort of effect. And studying this mutation might lead us to better understanding of how genetics will impact multiple systems in the body. And I have this little video to show you just because it's kind of cool. This is a bunny doing a handstand. Pretty neat mutation.
Okay, so for my This Week in Science presentation, I'm going to talk about how penguin hemoglobin has evolved to meet the oxygen demands of diving in a lot of the oceans around the world today. So basically, researchers have been conducting experiments and kind of comparing and they've been comparing the gene sequences of ancient penguins to the gene sequences of modern penguins to kind of see how the evolution of these species genetics has kind of adapted to take into account the different oxygen demands in different areas of the world. So basically, through these experiments and through these gene sequencing of modern penguins and their ancient counterparts, they found that that relative to land-dwelling birds, such as just all the birds you see driving around or in the forest, that penguin blood contains more hemoglobin. And for those of you who don't know what hemoglobin is, it's basically just the protein in our blood that picks up oxygen from the lungs and transports it through the bloodstream and drops it off at and kind of exchanges oxygen in the lungs and drops off at various tissues. So basically what these researchers found is that a hemoglobin abundance could partly, not fully, but partly explain the underwater proficiency of penguins who dive deeper than any other bird. And just an example of this is emperor penguins have been documented holding their breath for over 30 minutes while preying on krill, fish, and squid. So basically this finding that penguin blood contains more hemoglobin than did their ancient counterparts demonstrates not only how the experimental analysis of ancestral proteins and ancestral gene sequences can reveal the mechanisms of adaptation, but also that organisms are able to make these physiological adaptations in response to new environmental challenges and in response to just a dynamically changing world.
Okay, so last but not least, I want to talk about what people are calling zombie genes. So basically, data collected from fresh brain tissue, which was removed during like a brain surgery. Like sometimes they remove tissue for like strokes and that kind of stuff. So they were collecting data from this kind of tissue, where it's normally collected from tissue that's been sitting in like a tissue bank for a while. So it's interesting because it's like fresh, I guess. So they noticed that certain genes are expressed more after death than other genes, specifically in inflammatory cells called glial cells, which you have a little picture of over here. So they can grow appendages for many hours after death. You can see like the kind of little like branches that they grow over here. And this makes sense because their job is to clean up after like a stroke or oxygen deprivation, which are similar to the conditions that these cells will be experiencing after death. So this is important because it impacts research done on postmortem brain tissue, which is used to research certain diseases like anything from autism to like Parkinson's, that kind of stuff. So it's important because now we know that certain genes are expressed more, so we need to take that into account when we're doing this kind of research. Do you guys know what our code is? Or do we not have one? I can make one up right now.
Yeah, it's a Suez Canal, all lowercase, no spaces.
Sounds good.
Okay, thank you guys. Thanks for listening in. Have a nice rest of your day. Bye.